Diseases like Alzheimer's and Parkinson's can be caused by a kind of traffic jam in the brain. This occurs when the cells fail to get rid of dangerous substances and nerve cells die. Elin Esbjörner wants to figure this out in order to help those affected.
I want to understand why neurons die, because then we have better opportunity to develop new treatments that halt or stop the progression of the disease.
Just like in a city, different types of transport vehicles are required for a cell to function. These transport vehicles, called endosomes, are small biological packets that pick up substances and signals from their environment. A cell can also send out packets to communicate with other cells, which are appropriately called exosomes. The jobs that endosomes and exosomes performed are linked, and disturbance to their function can actually lead to disease.
I enjoy being out in nature, and then I feel that my mind can flow freely, and that I often think about my research and formulate ideas. For me, there's also a personal dimension, because I had an aunt that lived with Parkinson's disease. And it's a terrible condition with very few treatment options.
Det var ju som med min mamma, liksom, hon hade ju MSA, vilket ju oftast kallas Parkinson plus. Och då började jag ju plugga bioteknik. Jag såg
I find that my research on Alzheimer's and Parkinson's disease is very important, because there are so many that suffer. My interest in exosomes started in us wanting to understand how protein clumps transfer between cells. I started to ask colleagues that work on exosomes what I thought was very simple questions about what exosomes are and how they work. And I realized that there's a knowledge gap. Scientists haven't addressed these fundamental questions yet. And I'm very curious to do that. A major challenge with exosomes is that there are many different types. And I'm interested in understanding which types are involved in the transfer of toxic protein clumps in, for example, Alzheimer's disease.
Endosomes, and especially exosomes, are extremely small, making it difficult to even see them at all in order to study them. So the research team is now using a unique technology for advanced microscopy, which has been developed by Elin Esbjörner's colleagues at Chalmers. It consists of a small chip with nanochannels whose dimensions are so small that the exosomes can be studied one by one and their functions can be mapped. The goal is to label the different types of exosomes and follow their pathways between cells, what they contain, and what they transport.
För här kommer det verkligen en som är stor, och sen så där är en betydligt mindre exosom.
Elin Esbjörner has only been researching exosomes for a few years so far. But going forward, the new knowledge can be of great use for new methods of diagnostics and for medicines against neurological diseases in the brain.
We would like to study the entire transport chain that the exosome does, from the packaging of cargo and the release of the exosome from one cell to the transport and delivery of this cargo to the target cell.
If Elin's suspicions are correct that the exosomes transport protein clumps in the brain, it could lead to a new way of treating brain diseases such as Alzheimer's and Parkinson's. One goal for the researchers is to find exosomes that are good messengers, to then possibly use them to transport medicines. Then the exosomes would be able to be packed and deliver healing proteins, which could help a sick patient.
In the future, it may be possible to collect exosomes from a patient, load them with a drug, and create a personalized medicine. And this opens up possibilities for very cool treatments in the future.